The neurons, how they work. We'll look more at that since that's a little more complex. Almost all animals have a nervous system used for getting information, transmitting that to a central nervous system in most cases, and then processing that information and sending a signal in response. Again, we have the neurons themselves and the glial cells together making up the nervous tissue. Nerves interact quite a lot with the endocrine system. The response may be to start producing some hormone, for example. And so the nervous system sends a signal to the particular part of the endocrine system to start a hormone going in response to a particular nervous signal. So again, 4 basic steps here. First, you need to get a signal, the reception. Then that signal needs to be transmitted to where it's processed, the central nervous system in most animals. And then back out once a decision's been made to the effectors, the things that do something. In between the incoming and outgoing, you have to process that information, decide what to do. And then once that signal gets out, well, we need to actually do something about it. So the receptors, sensing, and the neurons that are either bringing that signal in or carrying messages back out are the peripheral nervous system versus the central nervous system, the brain, the spinal cord, that sort of thing that function in processing, making the decisions based on the information. If a neuron is getting sense data and bringing it in, it's a sensory neuron. If it is taking a message from the brain or spinal cord to a muscle and saying do something, it's known as a motor neuron. So the neurons send signals electrically along the cell itself. We have the cell body where the nucleus is. Most of the cytoplasm organelles that are basically keeping the cell alive. Obviously it needs that to function. But we have, again, 2 sets of branches. The dendrites, these shorter ones, that receive some sort of signal and bring it in. And then the long axon, which may branch, and then the tip often has branching, and that's carrying the signal on from the cell toward where it goes next, whether it's some other neuron or maybe a muscle, a gland that responds to that signal. These can be very long. They're quite skinny, but as cells goes, rather impressive in one dimension, their length. Again, we have branching. At the end, where the signal from the cell goes along the axon, reaches the branches at the tip, most nerves actually release a chemical, a neurotransmitter, and then that chemical will go through the gap between it and the next cell to cause a response in whether it's another neuron, a muscle, gland, whatever there. Synapse is the name for that gap. Larger axons often have special glial cells surrounding them, insulating, and with these present, the nerve signal travels faster. Also, if an axon is bigger around, the nerve signal travels faster. So we can have special structure in the axons to send a signal very rapidly. A nerve, in turn, is made up of lots of neurons together with protective connective tissue around them. The cell bodies often are clumped together in ganglia. Ganglia can function kind of like a miniature brain, an area where you have some signal processing before nerve signals travel on from there. Now, again, we think most about the neurons. They're the ones that are actually carrying the signal, but the glial cells are actually the majority of cells in the central nervous system. We have ones that provide support for the neurons, both physical and more figurative support in terms of carrying the nutrients to them, helping maintain the chemical balances. Some of these may function in neuron memory. Myelin sheaths, those insulating bits that help the signal travel faster, those are another glial type cell. We have special cells that are kind of similar to epidermal cells in that they are covering a surface, have some secretion function, various others there, maybe stem cells. And special ones, phagocytosis, eating waste bits, gobbling up bacteria, other things like that. These also help some of the signals. How does a nerve send a signal? Well, the cell membrane actually has electrical difference across them. Inside the cell is more negative, outside's more positive. To set up a difference like this, the cell must have used energy in order to pump more of some things in and other things out. But once it's done this, then that difference gives potential energy that can be used to do work, in this case, send a signal. Okay, just how different are the charges inside versus outside? The cell actually is pumping sodium out and potassium in, but it's pumping more sodiums out, so you get the polarity of more positive to the outside. And again, these are ions, electric charge, not going to mix well with those nonpolar lipid parts of the phospholipid membrane. So these do not move through the membrane on their own. They can only get through where the cell allows them with special pores and transporter proteins. Again, the cell has a special style of pump, brings in two potassiums, and pumps out 3 sodiums at a time. Okay, if we bring in +2, put out +3, net result is -1 on the inside. So the outside's getting more positive, inside more negative as the cell keeps up this pumping. Again, active transport's having to use energy from ATP to do this. Diagram here. So there are pores that can allow things just to go through passively, not active transport, but it does have the special pump protein using energy that pumps more sodium out and some potassium in. And you've got other things, chloride, larger proteins, things like that, that contribute to the difference as well. Okay, but some sort of signal comes along. Maybe electric, maybe chemical, maybe mechanical, like touch. But whatever this is, this can trigger the cell to open pores and allow the sodium back in. As sodium comes in, it's got a positive charge, so it's making the inside more positive. Was negative, getting more positive. Difference from outside to inside is becoming smaller. that's getting closer to making that neuron send a signal on, to firing. On the other hand, you could have stimuli that tell it to not do something. And so it pumps things the other way, makes it more negative, bigger difference, harder to fire the nerve. If enough stimulus comes in to reach a certain level, referred to as the threshold, then this triggers a sudden change as gates are opened in the transporter to proteins in the membrane. And this leads to electric signal traveling rapidly along the axon all the way to the other end, triggering the release of the neurotransmitter into the gap. So basically, the electrical charge inside versus outside changes very quickly, quite a lot. This ability is limited to nerve cells. Muscle cells can create a similar electrical charge difference, although they don't use it in the same way. And there are a few other cells that also make electrical differences like that, but not much.